Hello everyone, in this video we are going to learn about some widely dispersed topics which are relevant and just induces curiosity to learn them about more. So they are not connected at all, but still we will be learning them together just for curiosity. So first we are going to learn about the maternal inheritance. Maternal inheritance is a form of genetic inheritance in which certain traits or characteristics are passed down exclusively from the mother to her offspring. This type of inheritance is due to the presence of genetic material in the cytoplasm of the egg cell, which is contributed solely by the mother. The cytoplasmic inheritance is not subject to the normal rules of Mendelian genetics, which are based on the inheritance of genes located in the chromosomes. There are several examples of maternal inheritance in humans, some of which are associated with serious medical conditions. In this essay, we will be discussing about all these three examples about maternal inheritance in case of humans. So first is the Leber's hereditary optic neuropathy, which is shortly represented as HNON. Leber's hereditary optic neuropathy. Leber's hereditary optic neuropathy is a rare genetic disorder that affects the eyes. It is caused by mutations in mitochondrial DNA, which is inherited exclusively from the mother. LHON typically affects young adults, causing progressive vision loss and eventual blindness. Although LHON is a rare disease, it can have devastating effects in affected individuals and their families. Next is the mitochondrial encephalomyopathy, lactic acidosis, and stroke-like episodes, which is shortly represented as MELAs. MELAs is a mitochondrial disorder that affects multiple organ systems, including the brain. It is caused by mutations in mitochondrial DNA that is inherited exclusively from the mother. MELAs typically affect children and young adults, causing symptoms such as seizures, muscle weakness, and stroke-like episodes. Although there are currently no cure for MELAs, some treatments can help manage the symptoms. Maternally inherited diabetes and deafness. Maternally inherited diabetes and deafness is a rare disease of that type of diabetes that is caused by mutations in mitochondrial DNA. It is also associated with hearing loss, which is usually present from birth or develops in early childhood. MIDD is inherited exclusively from the mother and typically affects the adults over the age of 40. Although MIDD is a typically mild form of diabetes, it can have significant effects on affected individuals and their families. In conclusion, maternal inheritance is an important aspect of human genetics that can have serious medical consequences. The example of LHON, MELAS, and MIDD discussed in this essay illustrate the importance of understanding the mechanisms of mitochondrial inheritance and the potential risks associated with mutations in mitochondrial DNA. Ongoing research in this field may eventually lead to new treatments for this and other mitochondrial disorders. Next is the sister chromatid exchange. Sister chromatid exchange is a genetic phenomenon that occurs during the process of DNA replication in eukaryotic cells. SCE is the exchange of genetic material between two sister chromatids that are produced during the process of DNA replication. The exchange of genetic material between sister chromatids is an important mechanism of genetic variation and can also provide information about the effects of environmental factors on DNA damage and repair. The process of SCE occurs during the S phase of the cell cycle. When DNA replication takes place, each chromosome is duplicated, resulting in two identical sister chromatids that are connected at a region called the centromere. During SCE, the two sister chromatids exchange genetic material at specific sites along the length of the chromosome. This exchange results in the formation of new genetic combinations which can be passed on to the daughter cells during cell division. The frequency of SCAs can be affected by various environmental factors such as exposure to chemicals or radiation. Studies have shown that exposure to certain chemicals or radiation can increase the frequency of SCE, indicating that such factors can cause DNA damage that requires repair. Other factors such as aging and disease can also affect the frequency of SCE. SCE can be visualized using a variety of techniques such as chromosome staining and microscopy. One common method is the use of bromodioxyuridine, a chemical that is incorporated into the DNA during replication. By exposing cells to bromodioxyuridine, And then staining the chromosomes, researchers can identify the sites of SCE. SCE has important implications for both genetics and medicine. For example, SCE can provide information about the genetic diversity within a population and can be used to study the effects of environmental factors on DNA damage and repair. SCE can also be used to identify individuals at risk for certain genetic disorders such as Bloom syndrome and Fanconi anemia, which is associated with the increased frequency of SCE. In conclusion, cystochromatic exchange is an important mechanism of genetic variation that occurs during DNA replication in eukaryotic cells. The exchange of genetic material between cystochromatids can provide information about the effects of environmental factors on DNA damage and repair and can be used to study genetic diversity within populations. The visualization and study of SCE has important implications for both genetics and medicine, including identifying individuals at the risk of certain genetic disorders. Next, we will be learning about the interrupted genes. Interrupted genes, also known as split genes or discontinuous genes, are genes that are interrupted by non-coding regions of DNA called introns. The coding regions of interrupted genes, known as exons, are separated by introns, which can be removed during the process of RNA splicing. This process allows different combinations of exons to be joined together, resulting in a wide variety of different mRNA molecules that can produce different protein isoforms. The biological effects of interrupted genes can be significant. For example, mutations that affect the splicing of introns can result in abnormal protein isoforms that may be non-functional or have altered activity. Additionally, mutations in the regulatory regions of introns can affect the efficiency of splicing, leading to altered protein expression. Interrupted genes can be found in many eukaryotic organisms, including humans. One well-known example is the gene encoding the human blood clotting factor 8. This gene is interrupted by 26 introns, which is removed during the process of RNA splicing to produce a functional mRNA molecule. Mutations in this gene can result in various forms of hemophilia, a bleeding disorder caused by deficiency in clotting factors. Another example is an interrupted gene is the DMD gene, which encodes dystrophin, a protein important for muscle function. The DMD gene is interrupted by 79 introns, and mutations in this gene are associated with Duchenne muscular dystrophy, a severe muscle wasting disease. Interrupted genes can also play important roles in the evolution of organisms. The presence of introns can allow for the creation of new exons through the process of exon shuffling, which can generate new protein functions. Additionally, the presence of introns can allow for the accumulation of mutations without affecting the protein coding sequence, which can lead to the evolution of new regulatory elements or the creation of new genes through the gene duplication and divergence. In conclusion, interrupted genes are an important aspect of eukaryotic genetics. The presence of introns allows for the generation of a wide variety of protein isoforms through RNA splicing, which can have important biological effects. Mutations in the splicing machinery or in intronic regulatory regions can lead to altered protein expression or function. The study of interrupted genes are important for the understanding of genetic basis of many human diseases and for understanding the evolution of eukaryotic genomes. So gene families are groups of genes. Next topic is about gene families. Gene families are the groups of genes that share similar sequences and functions, often arising from gene duplication events during evolution. Gene duplication can result in the formation of new genes with altered conformations or expression patterns, which can provide an advantage in adaptation to changing environmental conditions. In this essay, we will discuss some examples of gene families and their functions. Homeobox genes. Homeobox genes are a highly conserved family of genes that are involved in the regulating developmental and cell differentiation in animals. These genes include transcription factors that bind to specific DNA sequences and control the expression of downstream genes. Homeobox genes are characterized by a conserved 180 base pair DNA sequence known as the homeobox, which encodes a DNA binding domain. The homeobox gene family is large and diverse, with over 200 members identified in the human genome. Mutations in homeobox genes can lead to developmental disorders such as holoprosencephaly and skeletal abnormalities. Hemoglobin genes are the next gene family. Hemoglobin genes are a family of genes that encode proteins responsible for transporting oxygen in vertebrate blood. The hemoglobin gene family has undergone multiple gene duplication events during evolution, resulting in the formation of different hemoglobin genes with distinct functions. For example, the alpha globin and beta globin genes are expressed at different stages of development and have different oxygen binding properties. Mutations in hemoglobin genes can lead to blood disorders such as sickle cell anemia and thalassemia. Next is the immunoglobulin genes. Immunoglobulin genes are a family of genes that encode proteins known as antibodies, which are involved in the immune response of pathogens. The immunoglobulin gene family is large and diverse, with different genes encoding antibodies with different antigen binding artifacts specificities. Antibodies are produced by B cells, which undergo a process of somatic recombination to generate a diverse repertoire of antibody genes. Mutations in immunoglobulin genes can lead to immunodeficiency disorders such as X-linked gamma globulinemia. Histone genes. Histone genes are a family of genes that encode proteins called histones, which are involved in packaging DNA into chromatin. The histone gene family is highly conserved across eukaryotic species, with different genes encoding different histone proteins with distinct functions. For example, the H3 histone gene is involved in maintaining chromatin structure, while the H2A histone gene is involved in regulating gene expression. Mutations in histone genes can lead to developmental disorders such as Cornelia de Lange syndrome. In conclusion, gene families are important for understanding the evolution of genes and their functions. The examples discussed in this essay demonstrate the diversity and importance of gene families in different biological processes such as development, oxygen transport, immune response, and chromatin structure. The study of gene families is important for understanding the genetic basis of many human diseases and for the development of new therapies and treatments. So, complementation tests. Now we are going to learn about the next topic, that is nothing else but the complementation tests. This is not related to the previous topic, but due to some reasons we are studying them together. These are the leftover portions from my syllabus, so I'm sorry for presenting all this in a one video, but pardon me for that. Complementation tests are a genetic technique. Complementation tests are a genetic technique used to determine if two mutations are located in the same gene or in different genes. This test involves crossing two individuals, each with a different mutation, to determine if their offspring show a wild-type phenotype. In this essay, we will discuss complementation tests in detail and provide examples on each cases of genes. Complementation tests involve creating a heterozygous organism with two different mutations. If the mutations are in different genes, the heterozygous organism will show the wild-type phenotype because the two different genes can complement each other. On the other hand, if the mutations are in the same gene, the heterozygous organism will show the mutant phenotype because the two mutations cannot complement each other. For example, consider two individuals with mutations in the same gene, gene A. These individuals are crossed and their offspring are observed for the wild-type phenotype. If the offspring show the wild-type phenotype, that indicates that the two mutations complement each other, meaning they are located in different genes. However, if the offspring show the mutated phenotype, it indicates that the two mutations do not complement each other, meaning they are located in the same gene. Complementation tests are commonly used in the study of human genetic diseases. For example, in cystic fibrosis, the genetic disorder that affects the lungs and digestive system, over 200 different mutations have been identified in the CFTR gene. To determine if two individuals with different mutations have mutations in the same gene or in different gene, a complementation test can be performed. If the offspring of the two individuals show the wild-type phenotype, it indicates that the mutations are in different genes. However, if the offspring show the mutant phenotype, this indicates that the mutations are in the same gene, so that they cannot complement each other. So another example of the use of complementation test is in the study of eye color in Drosophila melanogaster, the common fruit fly. Eye color in fruit fly is determined by two different genes, white and scarlet. Mutations in either gene can cause changes in eye color, but mutations in different genes can complement each other to produce a wild-type phenotype. To determine if the two mutations are in the same gene or in different gene, a complementation test can be performed by crossing two fruit flies with different mutations. If the offspring show the wild-type eye color, it indicates that the mutations are in different genes. However, if the offspring show the mutant eye color, it indicates that the mutations are in the same gene. In conclusion, complementation tests are a valuable tool in the study of genetics, allowing researchers to determine if two mutations are located in the same gene or in different gene. This technique has important applications in the study of human genetic diseases and in the study of model organisms such as Drosophila melanogaster. Now we are going to learn about the next topic, that is genes involved in apoptosis. Apoptosis, also known as programmed cell death, is a physiological process that allows cells to self-destruct in a controlled manner. This process plays a critical role in various biological processes, including tissue development, immune system regulation, and cancer prevention. Apoptosis is regulated by a complex network of genes that work together to control the process. In this essay, we will discuss some of the key genes involved in apoptosis and their functions. First is the BCL2 gene family genes. BCL2 family genes. The BCL2 family of genes is perhaps the most well-known group of apoptosis regulating genes. The family includes both pro-apoptotic and anti-apoptotic members. The anti-apoptotic members such as BCL2 and BCLXL. This is a new gene type, like BCL2 family genes. The BCL2 family of genes is perhaps the most well-known group of apoptosis regulating genes. The family includes both pro-apoptotic and anti-apoptotic members. The anti-apoptotic members such as BCL2 and BCLXL prevent apoptosis by blocking the activity of pro-apoptotic proteins. The pro-apoptotic members such as BAX and BAC promote apoptosis by disrupting the mitochondrial membrane and releasing cytochrome C. So we can say BCL2 family genes are of two types. First type is the anti-apoptotic members. For example, BCL2 and BCLXL. This prevent the apoptosis process by blocking the activity of pro-apoptotic proteins. But the later stage, which are promoting genes like pro-apoptotic members, they promote the apoptosis. For example, BAX and BAC. Caspase genes. Caspases are a family of proteases that play a critical role in the execution of apoptosis. They are activated in response to various stimuli, including DNA damage and cytokine signaling. Caspases cleave various cellular proteins, leading to the characteristic morphological changes associated with apoptosis, such as cell shrinkage and DNA fragmentation. Tumor protein P53 gene. The tumor protein P53 is a transcription factor that plays a crucial role in the regulation of apoptosis. In response to DNA damage, P53 is activated and induces the expression of pro-apoptotic genes such as PAX. P53 also represses the expression of anti-apoptotic genes such as BCL2, leading to an increase in the pro-apoptotic and anti-apoptotic ratio. So there will be an increase because of this tumor protein P53 gene. There will be an increase in the pro-apoptotic BCL2 portion. So that will lead to apoptosis. This is happening because DNA is damaged. So definitely that's it. Have to undergo apoptosis. Next is the FASL system genes. The FASL system is a cell surface receptor ligand system that plays a critical role in the regulation of apoptosis. FAS, also known as CD95, is a cell surface receptor that binds to its ligand, FASL. This binding triggers a cascade of events that leads to the activation of caspases and apoptosis. Defects in this system have been linked to autoimmune diseases and cancer. Next is the NFKB gene. The NFKB gene is a transcription factor that plays a critical role in the regulation of apoptosis. NFKB is activated by various stimuli, including cytokines and oxidative stress. In response to activation, NFKB induces the expression of anti-apoptotic genes such as BCL2 and BCLXL, leading to the decrease in the pro-apoptotic to anti-apoptotic ratio. So NFKB gene is a transcription factor that plays a critical role in the regulation of apoptosis. This induces the expression of anti-apoptotic genes, leading to a decrease in pro-apoptotic to anti-apoptotic ratio. In conclusion, apoptosis is a highly regulated process that is essential for maintaining cellular homeostasis The genes discussed in this essay are just a few examples of complex network of genes that regulate apoptosis. Defects in any of these genes can lead to pathological conditions such as cancer and autoimmune diseases. Therefore, understanding the molecular mechanism that regulate apoptosis is crucial for developing novel therapeutic strategies to treat these diseases. Which are the genes which we have just mentioned? BCL2 family genes, for example BCL2, BCLXL, and BAX and BAC. Caspase genes, tumor protein P53 gene, FASL system gene or CD95 gene and NFKB gene, CD95 or NFKB gene. Next topic, apoptosis in Sinorhabditis elegans, Drosophila, mammals and bacterial population. Apoptosis or programmed cell death is a process that is conserved throughout evolution, and it occurs in a wide range of organisms from bacteria to mammals. In this essay, we will discuss the process of apoptosis in 4 different organisms, namely Sinorhabditis elegans, Drosophila, mammals and bacterial population. So first, let's learn about apoptosis in Sinorhabditis elegans. Sinorhabditis elegans, a small nematode worm, has been extensively used as a model organism to study apoptosis. In Sinorhabditis elegans, apoptosis occurs during development and is regulated by a set of genes known as the CED genes or cell death abnormal genes. Apoptosis occurs during development as it is regulated by a set of genes known as CED genes or cell death abnormal genes. These genes include CED3, CED4 and CED9. The CED3 gene encodes a caspase protease that cleaves various cellular proteins to promote apoptosis. The CED4 gene encodes the adapter protein that activates CED3, and the CED9 gene encodes the anti-apoptotic protein that blocks CED4, thus preventing apoptosis. Next, Drosophila. Apoptosis also occurs in the fruit fly Drosophila, and it plays a critical role in various biological processes, including tissue development and immune system regulation. In Drosophila, apoptosis is regulated by a set of genes known as the Reaper family genes. These genes include Reaper, HID, and GRIM. These genes encode proteins that promote apoptosis by binding to and inhibiting anti-apoptotic proteins such as DIAP1. DIAP1 in turn inhibits the activity of the caspase protease TRONC, which is required for apoptosis. In case of animals, apoptosis in mammals is regulated by a complex network of genes including the BCL2 family of genes, the caspase family of proteases, and the tumor repressor protein P53. The BCL2 family of genes include both pro-apoptotic and anti-apoptotic members, and the balance between these two groups of proteins regulate apoptosis. The caspase family of proteases is activated by various stimuli, including cytokines and DNA damage, and they cleave various cellular proteins to promote apoptosis. The caspase family of proteases is activated by various stimuli, including cytokines and DNA damage, and they cleave various cellular proteins to promote apoptosis. The tumor suppressor protein P53 plays a critical role in the regulation of apoptosis by reducing the expression of pro-apoptotic genes and repressing the expression of anti-apoptotic genes. In case of bacterial populations, apoptosis-like cell death has been observed in bacterial populations and is believed to play a role in the regulation of bacterial population dynamics. In bacterial populations, apoptosis is triggered by a variety of stimuli, including nutrient depletion, antibiotic treatment, and oxidative stress. The mechanism of apoptosis in bacteria is not well understood, but it is thought to involve the activation of caspase-like proteases and cleavage of various cellular proteins. In conclusion, apoptosis is a conserved process that occurs in a wide range of organisms, from nematode worms to mammals and bacterial populations. While the specific genes and mechanisms that regulate apoptosis may differ between organisms, the fundamental process remains the same. Understanding the process of apoptosis in different organisms is crucial for understanding the role of this process in normal development and disease. Next topic is the mechanism of sorting and regulation of intracellular transport. Intracellular transport is the process by which molecules and organelles move within cells. Proper sorting and regulation of intracellular transport are essential for cellular function, as they ensure that molecules and organelles are delivered to their proper destinations within the cell. In this essay, we will discuss the mechanisms of sorting and regulation of intracellular transport, as well as provide examples for this process in action. So, mechanisms of sorting and regulation of intracellular transport. Vesicular transport. Vesicular transport is a process by which molecules and organisms are packed into vesicles and transported within the cell. This process is regulated by the docking and fusion of vesicles with the different membrane compartments, which is mediated by proteins such as SNAR, ARES, and RAB-GTPase. Next, motor proteins. Motor proteins are responsible for the movement of organelles and vesicles along the cytoskeleton. This process is regulated by the binding of motor proteins to specific cargo molecules, as well as by regulation of motor protein activity by other proteins. Membrane trafficking. Membrane trafficking is a process by which membranes and their associated proteins are transported within the cell. This process is regulated by the selective recruitment of cargo molecules to specific membrane compartments, as well as by the regulation of membrane trafficking proteins. Now let's learn about the examples of sorting and regulation of intracellular transport. Protein trafficking. Protein trafficking is the process by which proteins are transported from their site of synthesis to their final destination within the cell. This process is regulated by the selective sorting of proteins into vesicles, as well as by the regulation of vesicle fusion with specific membrane compartments. For example, the sorting of lysosomal enzymes into the lysosomes is regulated by the presence of specific targeting signals on the lysosomal enzymes, which ensure the selective diversity and delivery to the lysosome. Mitochondrial transport. Mitochondrial organelles that produce ATPs and critical for cellular function. Mitochondrial transport is regulated by motor proteins and move mitochondria along the cytoskeleton. In addition, mitochondrial trafficking is regulated by proteins that control mitochondrial fusion and fission, which are critical for maintaining proper mitochondrial fusion. Next, endocytosis. Endocytosis is the process by which cells take up molecules and particles from extracellular environment. This process is regulated by the selective recruitment of cargo molecules of specific membrane compartments, as well as by the regulation of vesicle fusion with specific membrane compartments. The selective uptake of low density lipoprotein is regulated by the presence of LDL receptors on the plasma membrane, which bind to LDL and initiate its uptake by endocytosis. In conclusion, the proper sorting and regulation of intracellular transport are essential for cellular function. This process is regulated to a variety of mechanisms, including vesicular transport, motor proteins, and membrane trafficking. Examples of this process in action include protein trafficking, mitochondrial transport, and endocytosis. Understanding these mechanisms is critical for developing new therapies for the diseases that result in the defects in these problems. So, mechanisms of sorting and regulation of intracellular transport. The sorting and regulation of intracellular transport play a critical role in cellular function and homeostasis. Intracellular transport is essential for the movement of molecules and organelles within cells, allowing cells to carry out their functions effectively. Proper sorting and regulation of intracellular transport ensures the molecules and organelles are delivered to their proper destinations within the cell, allowing cells to function properly. The importance of sorting and regulation of intracellular transport can be seen by protein trafficking. Proteins are synthesized into endoplasmic reticulum and transported to their final destination within the cell, such as the Golgi apparatus, lysosomes, or the plasma membrane. Improper sorting or regulation of protein trafficking can result in the localization of proteins, which can lead to disease. For example, defects in the sorting of lysosomal enzymes can result in lysosomal storage defects such as Tay-Sachs disease. Organelle transport. Proper transport of organelles such as mitochondria is critical for cellular function. Defects in organelle transport can result in a variety of diseases, including neurodegenerative diseases such as Alzheimer's disease and Parkinson's disease. In this disease, defects in mitochondrial transport and fusion or fission can result in mitochondrial dysfunction, leading to neuronal cell death. Next, Endocytosis. Endocytosis is the process by which cells take up molecules and particles from the extracellular environment. Defects in endocytosis can result in a variety of diseases, including hypercholesteremia, where defects in the uptake of low-density lipoprotein can result in elevated levels of LDL in bloodstream, leading to increased risk of cardiovascular disease. So that's happening in endocytosis. Signaling. The sorting and regulation of intracellular transport are critical for proper cellular signaling. For example, the selective transport of receptors to the plasma membrane is necessary for proper cell signaling. The defects in this process can result in a variety of diseases, including cancer. In conclusion, the sorting and regulation of intracellular transport is very important. So, when we just try to learn about the defects in sorting and regulation of intracellular transport, first, lysosomal storage disorders. Lysosomal storage disorder are a group of inherited metabolic diseases caused by defects in the sorting and transport of lysosomal enzymes. These enzymes are responsible for breaking down complex molecules in the lysosome. Defects in the sorting of lysosomal enzymes can result in the accumulation of the lysosome, leading to lysosomal shortage disorders such as Tay-Sachs disease, Gaucher's disease, and Niemann-Pick disease. Neurodegenerative diseases. Neurodegenerative diseases such as Alzheimer's disease, Parkinson's disease, and Huntington's disease are characterized by the progressive degeneration of neurons in the brain. Defects in intracellular transport, including the transport of organelles such as mitochondria and vesicles, can result in mitochondrial dysfunction and accumulation of toxic proteins in neurons, leading to cell death. Next one is the hypercholesterolemia. Hypercholesterolemia is a condition characterized by high levels of cholesterol in the blood. It can result from defects in the endocytic pathway responsible for the uptake of LDL cholesterol by cells. In familial hypercholesterolemia, defects in LDL receptor result in reduced uptake of LDL by cells, leading to elevated levels of LDL in the bloodstream and an increased risk of cardiovascular disease. Next, cancer. Defects in intracellular transport can also contribute to the development of progression of cancer. For example, defects in the transport of signaling molecules such as growth factors can result in the activation of signaling pathways that promote cell growth and survival, leading to uncontrolled cell proliferation and tumor formation. In conclusion, defects in the sorting and regulation of intracellular transport can result in a variety of medical conditions, including lysosomal storage disorders, neurodegenerative diseases, hypercholesterolemia, and cancer. How cancer is happening? Growth factors can result in activation of signaling pathways that promote the cell growth and survival, leading to uncontrolled cell proliferation and tumor formation. So, learning about all these problems can help in the development of new drugs and to treat these conditions. Next topic is microRNAs or miRNAs, small non-coding RNA molecules, typically around, it's non-coding and typically around 21 to 25 nucleotides long, that play a critical role in gene expression, regulation in various organisms. They were first discovered in the early 1990s in the nematode Cenorhabditis elegans, and since then it was found in many other species including humans. miRNA is transcribed from DNA sequences located within the genome, but unlike protein coding genes, they are not translated into proteins. Instead, they function as post-translational regulators of gene expression by targeting messenger RNA for degradation or translation inhibition. This process occurs through the formation of RNA-induced silencing complexes, which contain miRNAs and other proteins that recognize and bind to specific target mRNAs. miRNA play a critical role in numerous biological processes, including development, differentiation, and proliferation. They also have been implicated in various diseases, including cancer, cardiovascular disease, and neurodegenerative disorders. Dysregulation of miRNA expression can result in aberrant gene expression patterns and contribute to the pathogenesis of these diseases. miRNA can be classified into families based on their sequence similarity and target specificity. Members of the same family typically share a common seed sequence, which is a short sequence of nucleotides that is critical for target recognition. miRNA families are evolutionarily conserved, meaning that they are present in the multiple species and are similar functions across the species. miRNA can also involved in the complex regulatory networks in which multiple miRNAs can target the same mRNA or a single miRNA can target multiple mRNA. These networks can have a profound impact on gene expression patterns and cellular functions. The discovery of miRNA has led to the development of new therapeutic approaches for various diseases. For example, miRNA-based therapies have been developed for cancer, where specific miRNA can be targeted to either inhibit tumor growth or sensitize tumors to existing treatments. In summary, miRNAs are a class of small non-coding RNA molecules that play a critical role in the post-transcriptional gene regulation. They are involved in numerous biological processes and has been implicated in various diseases. The discovery of miRNA has opened up new avenues for therapeutic intervention and has shed light on the complex regulatory networks that govern gene expression in cells. So what is the importance of this microRNA? MicroRNA play a critical role in gene expression regulation in various organisms and have become an important area of research in molecular biology. This helps in the post-transcriptional regulation. mRNA play a critical role in the post-transcriptional regulation of gene expression by targeting mRNAs for degradation or translational inhibition. This process is essential for proper management, differentiation, and proliferation of cells and tissues. Next is the evolutionary conservation. miRNAs are evolutionarily conserved, meaning that they are present in multiple species and have similar functions across these species. This makes them useful for studying the evolution and biological systems in understanding the functions of genes across different organisms. Next, complex regulatory networks. miRNAs are involved in complex regulatory networks in which multiple miRNAs can target the same mRNA or single miRNA can target multiple RNAs. The networks can have a profound impact on the gene expression patterns and cellular function. Next, disease biomarkers. This regulation of miRNA expression has been implicated in various diseases including cancer, cardiovascular disease, and neurodegenerative disorders. This has led to the development of miRNA-based diagnostic tools and therapeutic interventions for these diseases. Next, therapeutic potential. miRNA-based therapies have been developed for various diseases. With specific miRNA can be targeted to either inhibit tumor growth or sensitize tumors to existing treatments. This has the potential to revolutionize the treatment of many diseases. When we just look at the functions of microRNA involve the post-transcriptional regulation of gene expression by targeting messenger RNA for degradation or translation inhibition. Gene expression regulation. miRNA regulates gene expression by binding to specific sequence in the 3' and translational region or UTR of target mRNAs, leading to translational inhibition or miRNA degradation. This process is important for a wide range of biological processes including development, differentiation, and proliferation. Next, developmental timing. miRNAs have been shown to play an important role in developmental timing in various organisms. miRNA have been found to regulate the timing of neuronal differentiation in the brain. Immune response. miRNA play a role in regulation of immune response by targeting genes involved in immune system function. Dysregulation of miRNA expression can contribute to autoimmune diseases and inflammation. Next, cancer. Dysregulation of miRNA expression has been implicated in various aspects of Dysregulation of miRNA expression has been implicated in various aspects of cancer development and progression. For example, some miRNAs act as tumor suppressors or inhibiting cell proliferation, while others act as oncogenes for promoting cell proliferation. Next, epigenetic regulation. miRNAs can also regulate gene expression through epigenetic mechanisms. For example, some miRNAs have been shown to target genes involved in DNA methylation and histone modification. Target genes through DNA methylation and histone modification. miRNAs are involved in post-translational regulation of gene expression and play a critical role in development, immune response, cancer, and epigenetic regulation. Thank you for listening.